Hello, everyone. Thanks for coming to this week's video lecture. Today, we're going to be going into Unit 3 and starting in Chapter 13, where we're going to talk about the microbiome of where microbes can be found on their body, the diversity that they have, and how they can interact um, with our body and how it can go astray and how some microbes can cause infection and disease. Um, and with that, you can find microbes all throughout the body, not all throughout the body, but in many places of the body. And like on the skin, in the upper respiratory tract, in the mouth, in your gut, on your all parts of your skin. And this is what it makes up what is known as your microbiome and or your native flora. And each of the sites of your body are slightly different in the environment where there's different levels of moisture, different levels of sunlight and UV radiation, different concentrations of salt, um, different oxygen levels, different acidities. All of those things influence what kind of bacteria can live when and where on your body. Um, and so one place in your on your body, like in your ear, is going to look different than inside of your gut, for instance. And so there are efforts in the research community to characterize all of the different microbes and the diversity and the ecosystems that live on the body and in the different locations of the body. And so when you look at these locations, you'll find bacteria that don't cause disease, and these are typically considered as mutualistic or commensal, where if they're mutualistic, both us and them are benefiting from this interaction. And commensal is where, for the most part, they're benefiting, but we are just not being hurt in that case. And an example of this mutualistic behavior or mutualistic relationship is within our gut, where we consume nutrients and food, and that some of those foods that we can digest ourselves with our own enzymes, and other times we can't digest some things. And so the microbes are able to facilitate that digestion, and we can then share those microbes between us. And so we both benefit from that. Well, we're feeding them and they're helping us with that digestive process. And so there are places on the skin that are common to find bacteria. This is things like the skin, the respiratory tract, the digestive tract, the genitalia, and in the ear canals. But you also have places that don't have bacteria that are relatively sterile. These are things like your bones, your muscles, your like your spleen, your brain. Typically your blood is also sterile. And so these are the places that you don't want bacteria and or microbes. And when microbes or bacteria get into this, it can cause damage or issues and cause what is known as an infection, or if that progresses into a disease. And so these microbes are mutualistic, not just in the way that they provide nutrients and like vitamins and hormones and things in our gut, but they just kind of act as good neighbors. So there's something called microbial antagonism, and this is like if you can think of your skin as a place that bacteria live, this is an ecosystem that bacteria colonize and they live. And it is made up of different enzymes, sweats, salts, and oils and hair and mechanical stresses when people move things around, soaps and things like that. All of those things define this environment or ecosystem for this bacteria. And this bacteria live in this place and they take up space, they take up the nutrients and all of those things. And so when you have a pathogen and they try to colonize that same location, there's already bacteria in that location. They're already eating the stuff that's available. They are already native to that environment. And so they understand, they don't understand anything, the bacteria, but like they already are established in that place, and there's so many of them, right? They're covering it. So when a bacteria comes in, they have to compete for space, they have to compete for resources, and they have to be adapted to that environment. And so this idea of microbial antagonism is when they're trying to compete for that space and that nutrients that the native microbiome provides, and is kind of providing a layer of protection from these or like competition from these pathogens. And so when you look at the skin, you have things like we talked about. You have oil glands that are putting oil onto the surface. You have hair, you have mechanical stress, you have sloughing off of skin. That's so the surface is constantly being replaced at the epidermis. You have sweat glands, and that has high concentrations of salt. You have enzymes that are being produced in these oil glands, and hair follicles that are being produced out and onto your skin. And so all of this area is colonized by different microbes that make up the microbiome of your skin. And the different in your skin could have different composition, like your microbiome on your skin could have different compositions in one place and another as well. Right. Some places are much more damp, or some places are much more anaerobic, where there's less oxygen. And so um So yeah, so bacteria live in these, can get into these glands and these hair follicles. If you get an infection in these hair follicles, you'll get like swelling and like pustules, like you get like gazette or whatever. Um, in the respiratory tract, we'll talk about that later, I guess. But in the gastrointestinal tract, you have the mouth, you have the pharynx, and the esophagus, which are all colonized by microbes. And then you have your stomach, which has stomach acid, which is able to kill a large majority of the bacteria. There are some bacteria that can withstand the stomach acid, but for the most part, you don't, it's typically considered a sterile environment. Um, and this is where a lot of bacteria that you eat is going to start to die. And this is a barrier for entry into your body. And then we'll talk about that in a different chapter. As you go through your small intestines, there's typically less bacteria. Some reports are showing that there might be some bacteria growth in there. But that's still all being researched. You have the large intestines, which is colonized by large amounts of bacteria and different species, and they aid in metabolism and things. And then you have so in that digestive tract, you have Neisseria species, Lactobacillus, Bacteroides, and fungus. You have Escherichia coli, Enterobacter, Citrobacter. Those are going to be helping you with the vitamins. Clostridium, Bacteroides, and Lactobacillus, that would help with lactose fermentation and digestion. You have the respiratory tract, where you have the nasal passage, which is not a sterile environment. It has mucus and things like that, which help protect and control and define what that environment looks like and feels like, right? Where it has high concentrations of oxygen. It's a very damp environment, and it has mucus. So it's it has some interesting barriers and like topology or environment to live in. And so certain bacteria are specifically keen or very good at living in those regions. As you go down the respiratory tract, you have cilia and things that start to clear out pathogens and microbes. And so you start to get into a more sterile environment down into the bronchioles and in the lungs. You then have the genitals, where you have the outer outside of the genitals, which is just obviously exposed to the environment and has its own microbiome. But you also have the beginning of the urethra, which can be colonized with some bacteria, as well as vaginal openings, and the anus also have colonies of bacteria. And again, the environments within those regions are different, where the anus is more of a anaerobic environment, and same with the vaginal canal. And those are going to define these regions. And there's different pHs in these regions as well that are going to define what different bacteria can and can't live in these locations. And when pathogens can make it through and actually colonize these places that they're not supposed to, or into sterile tissues that they're not supposed to, they start to establish infections and cause disease. And when these pathogens cause disease pretty much solely, where that is pretty much the only interaction they have with humans, they are called true pathogens. And these are things like influenza virus, Ebola, things like that. And then you have the opportunistic pathogens, malaria. You have opportunistic infections, where these are things that are going to take advantage of the opportunity to colonize your tissues and wreak havoc. But they don't do it readily. You don't see it in all people. A lot of this time is because the immune system is very good at clearing them away, or it just takes a lot of that bacteria to get inside of you to establish that infection. And this is something like Serratia marcescens, for instance, is one of those. And so there are different aspects, different factors that can contribute to how easy it is for a pathogen to get inside your body and to establish an infection. And these things are factors like age, genetics, your immune system, autoimmune diseases, as well as other characteristics like your current and past infections. And if you're going through specific treatments with medicines that can hinder your responses, cellular responses like chemotherapy. So you have what are known as virulence factors. So you have these factors that are on the host side that are impacting the severity of the infection, but you also have factors on the pathogen side, and these are called virulence factors. And these are things like toxins or antibiotic resistance, or different metabolic pathways or alternative metabolic pathways that can make a bacteria just like have a few edges in or a few, I was going to say edges, but that doesn't make sense without the context of like when you're in competition and you have the edge on someone. Virulence factors give the pathogen the edge on their host, essentially. And so these things are like toxins that help break down cellular tissue to make a better environment for the pathogen. Or these things could be like antibiotic resistance, so it can help resist treatments. But a bacteria or a fungus or a virus, I tend to say bacteria just because of habit. But when it starts to try to find these regions that it's not supposed to be in, whether it's a sterile tissue or a tissue that it's just not supposed to be colonizing, it has to get into the body or get into that region. And this is what is known as the portal of entry. And this can be like a break in the skin. This can be inhalation through your respiratory system. This can be through eating through contaminated food or water. This can be through sexual intercourse. This could be through breaks in the skin. But however it gets into your body, you have to have that bacteria get in there, and then it has to try to find this place that it's going to establish this infection. And it's going to go through a phase called attachment. And this is where it firmly attaches into the tissue so that it's not easily flushed off by moving interstitial fluids or through mechanical stress. So like if you got a fungus here and I could just go, bye fungus, it's not going to make an infection, but if it can root itself in and hold itself on, even if I'm scratching at it, it's still holding on at least enough that it can keep growing, right? And so this attachment phase is using those cellular structures like those hair-like structures like fimbriae or the capsule to attach to the host tissue. And then once it's attached to it, it can start to replicate and divide through binary fission if it's a bacteria, or through viral replication if it's a virus. And then it's going to start creating many of them. It's going to start avoiding the immune system, and it's going to start causing disease. And this is that progression into an infection. And then eventually it's going to transfer to a new host, and it's going to leave that host through a portal of exit, which we're not going to talk about too much, but essentially that's how it leaves the body so that it can go infect another person through their portal of entry. And so a common route for this is like the fecal oral route, is where contaminated fecal material makes its way onto food or onto your hands and then onto your mouth. And then that person now has bacteria that would be in the gut now in the upper end of the digestive tract, right? In the mouth and the esophagus and things. And that can cause problems. And so, for instance, the mouth was the portal of entry, and then it'll go through, and then that portal of exit was the fecal route. And so that transmission was the fecal oral transmission route. But I'm pretty sure we learned that later on, not right now. So again, we have the portal of entry, and you can have those different ways of entering into the body. You can also get through the gastrointestinal wall, kind of, only in severe infections, truly. But that can also be a portal of infection. This would honestly cause like sepsis, right? So this would cause bacteria to get into the bloodstream, and then that would cause sepsis. So in this process of causing an infection, you have bacteria that are coming into the portal of entry. It's attaching into the tissue. It's trying to evade the immune system, but if maybe the immune system caught it, and so it's going to try again. And so how many bacteria or microbes are needed to go and try to establish an infection before an infection actually makes it to fruition? And this is what is known as the infectious dosage, which is the number of organisms that are needed to establish that infection through that particular route of infection. And so you have different, so you have so many different bacteria that have to get into that portal of entry, and they have to establish this adherence. We talked about how that adherence can be through fimbriae, or it could be through capsules. We didn't mention, but it also can be through receptor binding. And so, for instance, viruses have protein surface receptors, cell surface receptors on their capsule. This is a protein that can bind to the cell surface of our cells, and it can hold itself there, or it can cause it to envelop itself and infect the cell. But that can also be through molecular protein-protein interactions that can also be holding them together. Again, we talked about virulence factors. I guess I kind of jumped the gun a little bit on those. But we have, we talked about these virulence factors are the things that the bacteria have at their disposal to gain an edge to establish this infection. These are things that can destroy the tissue to make it more inhabitable for that bacteria. These can be things like exotoxins that are excreted out into the environment and digest the cells. This can be things like a capsule, which it helps to evade phagocytosis and opsonization, which is when you have antibodies that cover a bacteria or a molecule that can cover a bacteria that make it easier to swallow, and the capsule can help prevent that from happening. Here's an example of that where you have the capsule helping to prevent this phagocytosis within the phagocyte. Another thing that they have is Staphylococcus species and Streptococcus species have leukocidins. We can tell this is a protein because it ends in IN. And so this is a toxin that is excreted out into the environment, and it kills the white blood cells that would be working to clear this infection. These enzymes can be doing work and be remodeling the tissues to make it more apt for infection. So for instance, if you have a skin infection, it's going to be easier to be established in that site if you can really get down deep into the tissue. And so like, for instance, ringworms have keratinases that can dissolve and break down the keratin between your cells or inside your cells. And first off, that's why it's it sloughs off a lot. But also it helps it get down into the tissue more and establish and be better attached, essentially, right? And make a better infection. And you can have different kinds of enzymes. We can tell these are enzymes because they end in ase. And you can tell these are proteins because they have that IN, right? So it's mucin. So mucin is a protein, and ACE is a enzyme that can break down mucin. Keratin is a protein, and keratinase is an enzyme that can break down keratin. Collagen's also a protein. It doesn't end in an I. I don't know why on that one, but it's collagen is a protein, and then an collagenase can break down that collagen. So, and so it can break down like the joints between your cells to help get deeper into tissues. And you have exotoxins that are secreted out into the environment, but then you have endotoxins that are held inside the bacteria until cell death. And then once the bacteria dies, it releases what are known as lipopolysaccharides out into the environment, not like intentionally, but just because it's a component of the cell wall. We have essentially a reaction to this molecule. Lipopolysaccharides is a lipid and a complex branched sugar that are bonded together. And it elicits a very strong immune response. And this can cause things like chills, malaise, and shock. And so you can get very sick, and it can be fatal in some instances with having these endotoxins that are found within the cell wall of gram-negative bacteria. Something that's important to notice is that these toxins are excreted into the environment, or they're a part of cell debris after cell death. And so even if you kill all the bacteria or you get rid of all of that bacteria, doesn't mean that these toxins have been completely eliminated from the environment. And this is where food poisoning is very crucial, where even if you do kill the bacteria, you may still have released endotoxins within that food, and it's still going to make you sick and cause an immune reaction because it still has these toxins in it. They weren't able to be destroyed by whatever method you use to either sanitize, cook, or decontaminate those surfaces or food. And it's definitely something to think about. So these a model of these toxins, there is a type of toxin called an AB toxin model. And so it's essentially that there are bacteria that have toxins, and this is a common like configuration for these toxins. But each bacteria kind of have, not each bacteria has, some bacteria have their own AB toxin like analog or like a similar system to this. Sorry, my English isn't very good, apparently. So what it is it's called an AB toxin, and it's these two protein subunits that are joined together by a disulfide bond. And when the B portion binds to the receptor of the cell that it was wanting to target, this causes endocytosis, or an enveloping of the toxin into vesicles. And these vesicles typically like maybe turn into endosomes and get really acidic, and or there's some enzyme that cleaves, something happens, it breaks the bond between the A and B. And this causes this A toxin to be released into the cell, and it can go on and do some kind of signaling and some kind of activation and cause some kind of havoc that's not supposed to happen. And this is how some toxins have a mode of action and affect the cells in your body. There are some fundamental differences between endotoxins and exotoxins, where endotoxins are found within the cell wall and exotoxins are excreted out to the environment. Exotoxins can be easily denatured, but endotoxins are a lot more stable. And that's because they're lipid and polysaccharide, and they're not proteins. Proteins are neatly folded, and so they're held together by weak forces, while these endotoxins are covalently bound together, and so they're held together by strong bonds, just like any other chemical. While the protein is essentially, if you looked at it in the same way as it's just a chemical, it's just one long string or strand. But because of like hydrogen bonds, van der Waals forces, and disulfide bridges, it forms into a complex 3D structure that can do some functioning, right? We talked about that in the last lecture, I think. And so, right. So, yeah, let's talk about once these bacteria have established this infection, the person clinically is going to start experiencing this infection and start understanding that something is going astray. And so when a patient is able to describe their experiences, how they're feeling, what they're experiencing, these are what are known as symptoms. And these are things that are subjective and objective to the person. And in the clinic, you have what is known as signs, and these are things that are objectively measurable. These are things like your blood sugar. These are things like your temperature. These are things like your height and weight. These are things like the number of bacteria that they have would be a sign. In like a harsh, very like strict definition, but like in practice, that's probably not what they talk about with signs, but signs are things that you can measure in the clinic. They are just true. While symptoms are the things that the patient experiences. And when you take them together, you can understand the presentation of different infectious diseases and how they present differently. And you can then understand, well, this disease or this bacteria has these signs and symptoms, which are classified as a syndrome. And that's how we can work to diagnose these different diseases. And so you can describe an infection depending on where it's located within the body. If you have a specific and singular location, it's called a localized infection. If it's found all throughout the body, like a sepsis, this would be called a systemic infection. And if it's something like cancer and then it's metastasized, this is something that's called like a focal infection. However, in this case, we would be talking about something that would be like you have an upper respiratory infection, and then it localizes down into a lower respiratory infection, then maybe it turns into sepsis. That would be the progression of that would be turning from a localized infection in that upper respiratory system. You would migrate that into a focal. I don't know if it would be considered focal, honestly, at that point, but eventually it would turn sepsis, and at that point, it would be a focal infection. These infections can be a single bacteria or a single microbe, and or it could be a whole host of different microbes. And part of the reason you can get a whole host of these microbes would be something like a stab wound, or maybe you have a tear in your gut lining, and then you have like your gut contents leak out. And so all of that bacteria gets into your abdominal cavity. And so it's just all different species in there causing an infection. Or you have a specific bacteria that got in where it's not supposed to, it proliferated very well, and it became a singular infection. And so those are the two kind of, you can have a mixed infection or you can just have like a regular infection. When you have that first, sorry, I have functional movement disorder. My brain functions, forgets. Sorry, my brain forgets how to function sometimes because of a functional movement disorder. Oh, goodness. So you can have one bacteria infection, or you can have a mixed infection. Typically, you're not going to say you have one bacterial infection. You're just going to say you have an infection, and people are going to assume that it's just one bacteria or one microbe. Mixed infections, you would just very specifically state that is happening. And so sometimes you have progressions of a infection or waves of infection where you may get an infection, it weakens the body to the point where it allows for a second infection to come in and create a secondary infection. This is kind of similar to like having an upper respiratory infection and then it turning into a lower respiratory infection. But in this case, we're talking about, for instance, maybe you have a respiratory infection that lowers your immune system, and then you're able to get or maybe you have like chronic pneumonia and that lowers your immune system. And so then you start getting sores on your skin that you can't heal because your immune system isn't able to clear those wounds. So that first set of infections is called the primary infection. And these are the infections that are going to essentially be like the things that set things into motion that are going to weaken your body to, if you can't resolve that infection, it could lead into a secondary infection. Sorry, my brain is going wild right now. Or it feels like it. I usually rewatch the videos and I'm like, it literally seemed fine. So I apologize, but I did want to at least let you know what's happening. So these secondary infections are typically called opportunistic infections. And so you have these primary infections and then it weakens your immune system, or it causes the environment to be more favorable, right? It changes the environment and causes tissue damage. Now it's easier for a bacteria that's just not usually strong enough to outcompete the typical microbiome to take hold. And so this is where the secondary infections or opportunistic infections come into play. When you have infections that are short-lived, these are called acute, but if they're long-lived and they occur over and over again, these are called chronic infections. The progression of these infections are depicted by this graph here, where you have an incubation phase. This is where bacteria are getting into you through the portal of entry. They're starting to attach into your tissue. They might be starting to divide. Eventually, they're going to start to cause tissue damage. Your immune system is going to notice, and you're going to start realizing that there's a problem. You're going to start feeling those symptoms, right? Those symptoms are those personal experiences of an infection. As you enter into the prodromal phase, this prodromal phase is when you start to feel sick. And then you get to what is known as the period of invasion, and this is where rapid infection is happening. Your cells are dividing not your cells, the bacterial cells are dividing rapidly. Large amounts of tissue damage is happening. This is where you're going to start, like an infection is going to start like sloughing off and infecting other people, or you're going to start infecting other locations in your body. And then eventually your immune system is going to catch up at the height of an infection, and it's going to start to resolve this infection, and your symptoms are going to start to be relieved under what is known as the convalescent period. And so we're going to talk about a few of the terms that you can describe as signs of infection, and these are measures of attributes of your body. And these are things like the measurement of your blood cells. This is like if there's present or not present bacteria in blood or interstitial fluid that is not supposed to be in. So for instance, leukocytosis is white blood cells, leuko, and cytosis would be lots of cells. So we have an increase of white blood cells within the blood. Penia is a decrease or the lack of. And so we have leuko, which is the white blood cells, and we have a lack of white blood cells. And so we have a decrease of white blood cells in the blood. And so this would be if we had no white blood cells or little, and this would be if we had a lot. You have sepsisemia. I don't know what the root words of these ones are, but it's essentially sepsis, which is the growth of bacteria or microbes within the blood that is causing an infection. And depending on what the microbe is, you could call it bacteremia or viremia. And this emia is something you'll see with blood a lot. So you can have people that are infected or can infect others and not be symptomatic, and they can these are what are known as asymptomatic infections, where they don't show symptoms, but they might have some signs, but they don't show or feel sick themselves. And so most of the time these are unreported because they don't go to the doctor when they don't feel sick. When these bacteria start to get into the body, that is known as a latency period. During that period of incubation, right? So we have that period of incubation right before the prodromal phase, before that infection is kickstarted. That incubation phase or latency period is the time it takes for that bacteria from getting into your body to start creating that infection. And remember, you have to have a certain amount through that infectious dose to establish an infection. And when you have a chronic infection, you're going to always have that bacteria or microbe in you. And so you'll be known as what is called a chronic carrier. And at times, chronic carriers are able to shed these infectious agents to people around them. And when you have these long-standing infections, a lot of the time they're causing permanent or long-standing remodeling to the tissues that are causing damage. This is called sequelae. And so we're talking, yeah, sources and transmissions. And so this is how our bacteria get to these portals of entry or the site of infections. So microbes and bacteria all have a place that they call home. And these are what are known as the reservoirs. And these are the essentially, these are like soils, these are water, these are the air, this is your skin, this is your gut. This is essentially where you're going to find this bacteria primarily, and what environment is it most adapted to living in. Again, this is called the reservoir. And so you can either get these pathogens directly from where they live through these reservoirs, or you can get them from a secondary object. And these are what are known as sources. And we have specific names for these sources, depending on if it's an animal or if it's an object. You can have what is known as a carrier, which are, we'll have what is a vector and, yeah, a vector and a carrier. So you have a carrier, which are like these biological I don't think carrier is it. It's vector and there's another one. Sorry, give me a sec. Fomite. It's a vehicle. That's what it is. Knew I wasn't crazy. So anyway, so a carrier are when we're talking about people and we're talking about how bacteria move between people, we're going to talk about them as carriers, where there are carriers that are infected and they are just asymptomatic, or they are symptomatic and they're just shedding infectious agents. And these are depending you can have carriers that are, when they're sick, they're just spreading their disease, or you can have asymptomatic carriers that are, depending on their stage of infection, you can have incubation carriers, convalescent, or chronic carriers, depending on what stage of infection they're at. But for the most part, you can just consider them asymptomatic, where they're not showing symptoms, but they're able to pass that infection on to other people. You have passive carriers, which these are going to be the things like in nosomial infections, in infections that are acquired in medical facilities. I'll say the word in a minute. These are oh my gosh. Sorry, my brain is just. Okay, so we have a passive carrier who's not infected in any capacity and is not going to get sick, but they are the person who carries it from a sick person to an unsick person who will get sick. Sorry. We then have vectors, and these are going to be animals and that are involved in the transmission of microbes from their reservoir to a person to be infected. And depending on if that organism is directly involved in the life cycle of the pathogen, it would either be called a biological vector or a mechanical vector, where mechanical vectors are going to be these organisms that are just letting these pathogens hitch a ride and not just grow inside of them. And so you would have something like a like Yersinia pestis or the Black Death on rats for instance, would be a mechanical vector, but for instance, Toxoplasma gondii, which is a toxoplasma that can infect mice, humans, and cats, and each of those hosts have different cell life cycles and stages, and those would be considered biological vectors. When a infection, when a microbe can infect humans from a animal, when a microbe can infect an animal and a human both, this is what is known as zoonosis. And typically it's where a animal infects a human. This happens in some cases with viruses and other diseases, but this isn't particularly dangerous because when it comes to public health, it's a lot harder to manage a wild population or a wild reservoir or a wild source of a disease than when it is completely strictly in humans, where you can do vaccination campaigns or educational campaigns to help deter the spread of a disease. Here are some examples of zoonotic diseases and their reservoirs and their mode of transmission, where you have mosquitoes that are helping to transmit different infectious agents. You have tick bites, airborne, and things like that. You can have reservoirs that are living, where the reservoirs are living things. Like here, you can see that hantavirus lives on rodents. But we also have non-living reservoirs like the soil, water, and air that where like Clostridium botulinum, Clostridium anthracis, all kind of live, or it doesn't, give me a second. Sorry about that. And so then you also have Legionella and Giardia, which are found in contaminated waterways. And then you also have fungal spores that are found in the air. And so these diseases can be called communicable diseases when they can be transmitted from host to host or person to person, or they can be non-communicable, where they can only be infected from the source or the environment to a host, and then that host doesn't spread it beyond that person. You can have what is known as direct transmission, where someone spews or directly transmits a pathogen from themselves to that person. This can be through the fecal-oral route. Maybe they touch fecal matter and then they put it in your mouth or contaminated your food with it. Or there can be indirect contamination, which uses a secondary object. I apologize. If they touch their food, that would be a secondary object. So that would be an indirect transmission. My apologies. You'd have what is known as vehicles or fomites. Fomites are these inanimate objects that help the transmission of bacteria. And these are things like doorknobs. These are things like food. These are things like I think I said them all. And you can have indirect transmission, which are getting someone left a pathogen in the environment and then you picked it up from that environment. Or you can have direct transmission, which is from one person to another. You can have nosocomial infections, which are a class of infections that are acquired specifically in medical facilities. That was what I was trying to pronounce earlier. nosocomial infections, and they are these these infections that are acquired in medical facilities. Typically, you're already in an immune you're already in a weakened state typically, and so you're at high level of susceptibility to infection in these places. And then also there is higher rates of bacteria that that are harder to treat, like antibiotic resistant bacteria, like MRSA and things like that. And so you have to so these infections are particularly dangerous to a already at-risk population of individuals. And so it's important to put in fail-safes and hygiene practices to prevent this from happening. When you study these diseases in a population, this is called epidemiology. The government has established systems to help protect and study epidemiology. The World Health Organization and the Center for Disease Control and Prevention all work and monitor disease aspects, disease occurrence, infection occurrence, where and when they're happening, if they're happening at different rates, and if they need to be looked at as like if they're a concern or above normal. And they also just understand the baseline of what is a risk of where you're living. So for instance, one location in the globe might have some risks of infection that another place in the globe does not have. There's like, for instance, some locations that have a specific bacteria that is pathogenic. And so it's just aerosolized a lot easier. And so people can get sick easier. I don't remember what it is about, but I'm pretty sure it has to do with farms. But then they also have all of these programs that work to surveil these infections. I was talking about that. And so there are mandated reporting systems for hospitals to report these infections that are being monitored. And then the CDC puts out specific reports on what these cases look like and makes sure that it makes sure that this data is available for epidemiologists to study how this infection is working and monitoring the public health. When we look at these infections, you can look at them in a few different ways, where we have an endemic, which is a thing that is localized to a specific location for a long period of time. And so, for instance, this is something like culture is typically endemic to specific regions. You see it in specific regions, and it's there for a long period of time. You see this with specific diseases. This would be like I was talking about, there might be some specific farmland that just has specific bacteria in that makes that when it's aerosolized, it can cause infections. You can have sporadic, which are just found everywhere. You can have epidemic, which is when it is found when a specific infection and a disease is being found not sporadically, but at increasing levels across a region. And a pandemic is when you're seeing those levels of infection increasing in all parts of the world. And then sporadic is just it's found everywhere, but you're not seeing increasing cases. So that's the difference between sporadic events and epidemic is increasing frequency. And with that, that is the entirety of our lecture. So I would like to say goodbye. Thank you. Bye-bye.